Let me see. Oh, now it's better, right? Okay, so turn on. Great. So I'm Natalie Surkova, Radiology and Shared Resources in the Cancer Center. And this is my distinct pleasure to introduce our speaker today, Dr. Zaver Bhujwalla from Johns Hopkins. And I had the honor to know Zaver for almost 25 years now. So she's also not like formal mentor. She has been my role model and advisor. So Dr. Bhujwalla is the William Brody Professor of Radiology at the Johns Hopkins University School of Medicine. She serves as the Vice Chair of Research in Radiology and is the inaugural Director of the Division of Cancer Imaging Research. Dr. Bhujwalla work focuses on molecular and functional imaging in cancer, with a particular emphasis on the tumor microenvironment, cancer-induced cachexia, and theranostic imaging. And this is what she will be talking to us today. Dr. Bhujwalla holds joint appointment in oncology, radiology, and radiation oncology, and has directed the Molecular Imaging Center since 2003. She has received multiple highest awards from the Imaging Society, such as a gold medal for World Molecular Imaging and International Society of Magnetic Resonance in Medicine. She serves as the permanent member on many NIH study sections. She has received countless NIH R10 grants, P50 grants, P41 grants, and she has published over 250 publications in the area of metabolic and molecular imaging of cancer and theranostics. So before I give the mic to Dr. Bhujwalla, I want to give her a small token of our appreciation for coming here and giving us cutting edge research. Zaver, thank you so much. And this is for you from the Cancer Center. Oh, thanks.
Right, so good afternoon, everyone. And Natalie, thank you very much for the invitation to come and talk here today and for being such a perfect host. And it's just wonderful to be here and see this very impressive center that you have. And I'm just delighted to be here. And lots of old friends here too, like Dr. Kamel from Johns Hopkins, formerly. So it was great to meet up. So I'd like to talk today about some of the work we've been doing in our research program at Johns Hopkins. Here are my obligatory slides, so I have no disclosures, and these are my 4 learning objectives. So in my research program, we are very interested in 3 broad areas. So the first one, something we've been investigating for over 20 years, and that's the tumor microenvironment. Then more recently, we've become very interested in understanding how cancers affect the body, especially in the context of the syndrome called cachexia, which induces a profound weight loss and typically observed in pancreatic cancer, lung cancer, and some other cancers. And then we take the insights of what we gain from these studies and develop theranostic strategies. And theranostics is this term that's coined as a combination of diagnosis and therapy. And it's really this exciting new field which allows precision medicine of cancer. So like most academic centers, including yours here, and Natalie's been telling me of all the fantastic imaging equipment you have here, we have this multimodality imaging center. And most recently, we got a PET-MR scanner that allows us to do simultaneous PET-MR. We also obtained a optoacoustic scanner. Of course, we have multiphoton microscope, we have MR scanner, SPECT, SPECT-CT, ultrasound, etc. And these are examples of some of the images we can obtain with these modalities. So for instance, here, this is a PET MR image. And here we are looking at PDL1, the immune checkpoint PDL1 in a tumor model, and of course, overlaid with MR image. And you see this exquisite soft tissue contrast that gives you distribution of PDL1. So you see some in the lungs here, some in the spleen, and of course in the tumor. This is optoacoustic imaging of optoacoustic dye, ICG, in real time. So you can see the uptake of the dye in the tumor. We use our multiphoton microscope for things like intravital microscopy. This is something my colleague Dmitry Artemov has provided. We also do things like second harmonic generation microscopy to look at collagen fibers. This is an optical image to show metastatic dissemination in a mouse model. This is a map of vascular volume in red, overlaid with permeability in green. So we use MRI to quantify tumor vasculature. And this is a PET image, a PET CT image, where we've targeted prostate-specific membrane antigen. So this is a high PSMA-expressing tumor, low PSMA-expressing tumor, where we've delivered nanoparticles carrying small interfering RNA. And you can see that it's quite nice to be able to target the tumor. So we use all of these imaging modalities to answer the questions we have in our research program. And cancers, as you know, consist not only of cancer cells, but also have vasculature, lymphatic endothelial cells, collagen fibers, and then stromal cells like cancer-associated fibroblasts, macrophages. And all of these are collectively termed the tumor microenvironment. The tumor microenvironment is co-opted by cancer cells in their strategies to survive and escape treatment. Additionally, the abnormal vasculature and lymphatics in tumors leads to areas of hypoxia, acidic and pH environments. And so at any given time, the tumor microenvironment is this fluid, dynamic landscape. It's with spatial and temporal heterogeneities, and imaging is really important to be able to follow these heterogeneities and to understand the role of these parameters in tumor progression and invasion and response to therapy. So in addition, cancers, of course, also you can view them, cancer, as a systemic disease, because we've been observing that cancers affect the body, and there's this constant exchange and interconnectedness with tumors and visceral organs or the brain, for instance. And again, imaging is really important to be able to view cancer holistically as this systemic disease and provide insights into improving outcome to treatment and also reducing morbidity. And then, of course, insights can be used for developing theranostic agents. So specifically in my research program, in the tumor microenvironment, we've been focused a lot on understanding hypoxia and its relationship to vasculature, understanding how hypoxia affects things like immune checkpoints like PDL1, the extracellular matrix like collagen fibers, fibroblasts. So those are the things we're working on in the tumor microenvironment. In the tumor macroenvironment, we are very interested in cachexia. And this is something actually that we were working with Dr. Kamel some years back with pancreatic cancer. So what we wanted to do is understand how metabolism changes in organs in the body with cachexia, and use that to come up with metabolic interventions that could slow down cachexia. And we picked pancreatic cancer because almost 80% of pancreatic cancers exhibit this syndrome, which is a major cause of morbidity and poor quality of life. And then for our theranostic strategies, we are primarily focusing on delivering nanoparticles decorated with different types of imaging reporters, depending upon the imaging modality we use, to deliver things like small interfering RNA to downregulate specific targets, including metabolic enzymes. And then together with Dr. Kobayashi at the NIH, we've also been implementing photoimmunotherapy, and I'll describe some of that work as well. So those are some of the areas we are working on, and I'll give examples from some of these areas in my talk today. So as you know, hypoxia is a major cause of resistance to therapy. It leads to aggressive phenotype, can promote invasion and metastasis. And because of our interest in hypoxia, we developed cells where the hypoxia response element is linked to a reporter gene. So you can either use green fluorescent protein, red fluorescent protein, or luciferase, so that when you take cells that express this cDNA and put them under hypoxic conditions, they express the reporter, as shown here, with green fluorescent protein. It can be red fluorescent protein or luciferase as well. And then we validated the system in vivo using a fiber optic probe to detect oxygen tensions. And here you can see that in this tumor, derived from these cells, there are these distinct areas that are fluorescing. So when we put the fiber optic probe in non-fluorescing regions, we see high oxygen tensions. In the fluorescing regions, we see almost non-detectable oxygen tensions. So we were able to validate the system to demonstrate that, yes, it does report on hypoxia. And this is just a similar thing with red fluorescent protein. And this is with luciferase, where if you inject luciferin, you get bioluminescence. And again, you can use it to detect hypoxia. So I'll just present a couple of examples with these reporter systems in our tumor microenvironment work. So we've been using MRI to quantify tumor vasculature. We use albumin GAD, DTPA, so it changes water relaxivity. And then we can come up with quantitative maps of vascular volume and permeability based on quantitative T1 maps following injection of albumin GAD. So as you can see, and this is a hallmark of cancer, that any parameter you measure is always heterogeneous. So here, typically you have this very heterogeneous vascular volume map with this very marked region which has low vascular volume and has high permeability. So we combined MRI and optical imaging using our GFP expressing reporting, hypoxia reporting cells. And what we were able to demonstrate is that, yes, indeed, these areas of low vascular volume have high GFP expression and are hypoxic, as you would expect. And these low vascular volume regions are also highly permeable, and that is most likely because hypoxia drives up vascular endothelial growth factor and increases permeability. Of course, this has implications for delivery as well, because it means that larger molecules like albumin GAD will not leak out from the tumor vasculature in well-vascularized tumor regions, but probably will leak out in these poorly vascularized, kind of doomed to die regions anyway. So this is an example of combined MRI and optical imaging. We next wanted to understand how hypoxia and collagen fiber distributions are related in tumors. So we thought all along that hypoxia would drive up collagen in tumors because hypoxia would lead to inflammation and we would see more collagen. Instead, when we used our tumor model, and here the hypoxia is shown by red fluorescent protein expression, we found that the hypoxic tumor regions actually had low collagen fibers as detected by second harmonic generation microscopy of collagen due to its non-centrosymmetric structure, whereas the non-hypoxic regions had high collagen. But around the hypoxic regions, we saw increased collagen deposits. So again, what we think is happening is that hypoxia drives up these degradative enzymes that break up the collagen. And in fact, we saw this in our cell perfusion system. So hypoxic regions, the collagen gets broken down. And so that is why we are not seeing high levels of collagen in these So with that, we come up with draining and pooling voxels. What we found is that the hypoxic tumor regions were, again, these silent zones where there was very little movement of macromolecules in the interstitium. So that made us think that these collagen fibers are actually mediating movement of macromolecules through the tumor extracellular matrix. And so we went to do water diffusion also and related to collagen fibers. And we found that the fractional anisotropy of the water, the directionality of the water movement tracks with the collagen fibers. So similar to what other groups have seen, where cancer cells use collagen fibers to track out of the tumor on their metastatic journey, we think that these fibers are mediating movement through the ECM. And then most recently, we've started to look at collagen fibers in stage 1 non-small cell lung cancers using the NIH's database. And here we find that primary stage 1 non-small cell lung cancers that go on to metastasize subsequently have much more collagen fibers, as shown here, compared to non-recurrent non-small cell lung cancers. This, again, is consistent with the possibility that these collagen fibers are being used by cancer cells to migrate out of the tumor to distant sites. So that's hypoxia and vascular volume and collagen. Now I'd like to present a couple of slides about hypoxia and the immune checkpoint PD-L1. So PD-L1, as you know, is expressed by cancer cells to escape immune surveillance. So we used our hypoxia reporting bioluminescent model to detect hypoxia. And then together with Sridhar Nimagada, we used a copper 64 labeled PD-L1 antibody to look at PD-L1 distribution. And what we found is that in the tumor, the regions that were hypoxic, as detected by bioluminescent imaging, showed increased PD-L1 expression. So we think that hypoxia drives up PD-L1 and contributes to escape of immune surveillance in tumors. And if you take cells and expose them to hypoxia, you see this increased PD-L1 expression. You see more PD-L1 expression around necrotic tumor regions, which are mostly hypoxic. And then we were able to show this in TCGA analysis of treatment-naive triple-negative breast cancer. Again, because of our interest in collagen and the extracellular matrix, and that is primarily put down by fibroblasts, we also wanted to look at how prostate cancer fibroblasts reacted to hypoxia in terms of PD-L1 expression. So to do this, we characterize PD-L1 in prostate cancer-associated fibroblasts and normal fibroblasts, and these are human prostate cancer fibroblasts and normal fibroblasts. And what we find, first of all, is that there's very high PD-L1 in prostate cancer-associated fibroblasts, but not in normal prostate fibroblasts. If you put the cancer-associated fibroblasts under hypoxia, you see an increase of PD-L1. Now, with our MRI systems, we have a way to look at the degradation of matrigel in a cell perfusion system. So we have a layer of matrigel and we do imaging, and we can visualize how cancer cells or fibroblasts degrade matrigel dynamically over time. So we took our prostate fibroblasts and we took our prostate cancer-associated fibroblasts. And as you see, the prostate fibroblasts break up matrigel in very quickly. Cancer-associated fibroblasts are not invasive, so they leave the matrigel pretty much intact. Under hypoxia, the prostate fibroblasts become even more invasive. The prostate cancer-associated fibroblasts leave the matrigel untouched. So we think that, at least with this model system, that the cancer-associated fibroblasts do not contribute to invasion in the tumor, but maybe the normal fibroblasts that surround the tumor are breaking up the matrix and allowing cancer cells to metastasize. But the cancer-associated fibroblasts are contributing to immune suppression by expressing PD-L1. And we could confirm this in vivo, where you can see that the fibroblasts are expressing more PD-L1 than the cancer cells. And also in human tissue microarrays, we see similar results. If we look at metabolism, so these are high-resolution spectra of tissue extracts, and you can see multiple metabolites. You can see that when you expose prostate fibroblasts to hypoxia, the hypoxic response is very different compared to the prostate cancer-associated fibroblasts. So I don't want you to go through all the metabolites, but you can see that many of these metabolites go up and many go down. So again, very distinctively different metabolic response of fibroblasts to hypoxia. So that's where we are with looking at hypoxia and PD-L1. And then, of course, we're also very interested in choline metabolism. And, many studies, including ours, have shown that cancers have altered choline kinase, as shown here. Choline kinase converts free choline to phosphocholine, as shown here, is always high in cancer cells and tumors, as shown here in a breast tumor model. And so one of the things we wanted to see is what happens to PD-L1 when you downregulate choline kinase. And again, we saw this seesaw effect where if you silence choline kinase, PD-L1 goes down. And if you silence PD-L1, choline kinase goes down. So again, there is this close interaction between the immune checkpoint PD-L1 and metabolism, and that can be used to uncover new treatments for more effective immunotherapy. And we've also now switched to mass spectrometry imaging, and this is a core now led by Christine Glund at Hopkins. And here we've started to characterize, together with Dr. Mike Goggins, normal prostatic tissue, benign prostatic tissue, and pancreatic cancer. And again, mass spec imaging, although it's not in vivo, it's ex vivo, you can see this beautiful spatial heterogeneity of your metabolite of interest. And you can start to relate it to pathology. So you can see, okay, what are the T cells here? What are the stromal cells, and so on. And so we've seen alterations in multiple metabolites, but notably in taurine, which is an immunometabolite, we see that it goes up in PDAC, and creatine is significantly decreased in PDAC. And again, these kinds of metabolic insights can help in coming up with novel treatments in pancreatic cancer. So speaking of pancreatic cancer, I'd like to come to cachexia, which is our second area. And cachexia, as I mentioned, is this syndrome that induces profound weight loss and is associated with poor quality of life, poor response to treatment, and can lead to significant morbidity and to mortality. And so some years back, Anirban Maitra gave us a panel of patient-derived human pancreatic cell lines, which we characterized, and we found that one of them, PA04C, mimics what is seen in patients in that it induces weight loss in mice. So you can see here that if you use a PANC1 tumor model, there's very little weight loss compared to non-tumor-bearing mice. But PA04C induces this significant weight change. And we initially started off looking at the brain in these mice. And here we found that we thought that the brain is a privileged organ. There wouldn't be change in brain weight, but we saw this significant decrease of brain weight. And of course, significant changes in metabolism. So we saw in the brain that in the cachexia-inducing model, there was a significant decrease of choline, an increase of glutamate, and an increase of formate. Dr. Goggins also provided us with human plasma samples from PDAC patients. So patients with stable weight and patients with weight loss. And similar to the change in glutamine in the mice in the brain, we found glutamate was significantly lower in the plasma of the patients with weight loss. So that led us to target glutamine metabolism in the PA04C cell line. So Balaji Krishnamachari, who's our molecular biologist, created PA04C cells expressing short hairpin RNA, either with glutamine synthase 1 and 2, glutaminase 1 and 2 downregulated, and with the glutamine transporter downregulated. So downregulating GLS1 and GLS2 had no significant effect on tumor growth, but downregulating the glutamine transporter resulted in this long latent period, after which the tumors did grow at a comparable growth rate as the empty vector wild type. But for us, what was very interesting was that for volume matched tumors, where you see that by 200 millimeters cubed, you see profound weight loss in these mice, the tumors derived with cells with the glutamine transporter downregulated had very little weight loss. So this opens up the possibility of targeting the glutamine transporter to attenuate cachexia in pancreatic cancer. Because we had the plasma samples, we also wanted to see if we could develop a screening method for pancreatic cancer. And so here, Mayapan Solayapan took these plasma spectra from malignant, benign, and normal participants, and we were able to then classify these groups of patients based on the neural network analysis of these spectra. So we're quite excited about this as well for screening methods. And then in this tumor microenvironment work, we're also looking using our optoacoustic scanner to look at brain hemodynamics. So here we've taken mice, again with PANC1, PA04C tumors and non-tumor bearing mice. We measure oxygen saturation under air and following oxygen breathing. And you can see that the mice with the cachexia-inducing tumors have a significant decrease in their response to oxygen breathing. So the blood vessels are not able to carry that additional oxygen in the brain. And so we think that the hemodynamics of the brain is also altered with cachexia. And more on this as we progress. And I had a really great discussion with Andrea earlier that this might be due to reduced RBCs. That is one of the side effects of cachexia. So very quickly, I don't expect you to look at all these metabolites. So we're also looking at visceral organs. So we see a decrease of heart weight, kidneys, liver, every organ we've looked at, there's weight loss. What I do want you to focus on is the spleen. So the spleen of all the organs, the spleen showed the greatest difference in metabolism. So again, this is an example of how cancers can affect visceral organs and visceral organ metabolism. So when we saw this change in the spleen, we thought, well, these are immune suppressed mice. What if you take syngeneic tumors? Do they also show changes in spleen metabolism? So we took 41, which is a triple negative breast cancer model in immune competent mice. We took E0771, also breast cancer model, and PANCO2, and we looked at the spleens. We found that these 5 metabolites are commonly altered, irrespective of the tumor model. So lactate, alanine, glutamate, glutathione, and glycine changed in the spleen consistently. So then we thought, well, if we are seeing these common metabolic changes, how do they affect T cells in the spleen? And so we looked at T cell exhaustion, and this is work done by James Barnett, my PhD student, and we found that there is significant T cell exhaustion in the spleen with tumor growth. And again, this raises the question of whether these T cells are exhausted before they even reach the tumor, again, providing insights for immune response to cancer and immunotherapy. So with this in mind, I'll come to the last few slides of my talk, which is Theranostics. So Theranostics, as I mentioned before, is a combination of diagnosis and therapy. And typically what is done is you take a nanoparticle. Recently, there's a lot of interest, for instance, in radiotheranostics, where you put an alpha particle or beta particle on something that's targeted to your receptor antigen of interest. So we take nanoparticles, we target them to a specific component, and they are loaded with some therapeutic cargo. In our case, we use small interfering RNA. And we deliver them to some specific aspects. So you can target them to PSMA, you can target them to FAP alpha if you want to target fibroblasts, you can target them to PDL1, for instance. And so they release their cargo at the target of interest. And so we've been kind of switching to photoimmunotherapy for theranostics. And photoimmunotherapy is something that Dr. Kobayashi at the NIH found that if you take this near-infrared dye, IR700, and you conjugate it to an antibody that binds to the receptor or antigen of your interest, so it goes and binds there. And then because IR700 is fluorescent, you can detect its binding by optical imaging. Then if you shine a therapeutic dose of light, the IR700 dye absorbs the light energy and re-emits it at the cell membrane and makes holes in the cell membrane and kills the cell. So you get very target-specific cell death. And so with that in mind, we thought, can we use it to target myeloid-derived suppressor cells in the spleen? And so to do that, and MDSCs, as you know, are a major source of immune suppression, poor response to immunotherapy, increased aggressiveness, increased invasion, metastasis. So we targeted MDSCs by using the GR1 antibody that is expressed by mouse MDSCs. And here you can see that it very nicely goes and localizes in the spleen because these mice have the 41 tumor growing that causes an expansion of MDSCs in the spleen, which is why you see that the spleen light up. And now if you expose the spleen to therapeutic dose of light, the light gets absorbed, the dye breaks down, it stops fluorescing. So you see that the fluorescence is reduced, and you can basically kill the MDSCs in the spleen. And then we looked at metabolism in the spleen. So if you remember, I showed you those 5 metabolites that are consistently altered in the spleen earlier. So now this is the 40 model. These are non-tumor bearing mice and spleen metabolism from non-tumor bearing mice. So we get this by high resolution spectroscopy of spleen extracts. So these are like exposed to photoimmunotherapy, to no photoimmunotherapy, not injected, and non-tumor bearing. So non-tumor bearing mice have, as shown in this heat map, a very specific metabolic profile. Tumor bearing mice have a different metabolic profile in the spleen. If you inject the antibody with the IR dye, but don't expose the spleen to light, the metabolic profile is the same as the tumor-bearing mice. Now, if you expose the spleen to photoimmunotherapy, the metabolic profile becomes similar to that of the non-tumor-bearing mice. So that is most likely because the MDSCs are getting killed and you're getting a metabolic profile comparable to the non-tumor-bearing mice. So now we want to see if there's less T-cell exhaustion following photoimmunotherapy, and those are ongoing studies. I mentioned that we can also deliver small interfering RNA. So we've done this for PDL1, as shown here. Again, you take a nanoparticle. Here it's a dextran nanoparticle with a bonds that release small interfering RNA under acidic pH environments. We were able to show downregulation of PDL1. You can do the same thing with choline kinase, show decrease of total choline in the tumor. And here we used it for PSMA-specific siRNA delivery, where we delivered 2 different siRNA for PDL1 and CD46. And again, we're able to show that we could achieve target gene downregulation in a PSMA expression specific manner. So just examples of Theranostics. So I'd like to conclude here and see if anybody has questions. But just to conclude, I hope I've demonstrated that imaging has really an important role to play in understanding the tumor microenvironment and in understanding cancer in all its complexities and heterogeneities. And imaging, of course, is really valuable in viewing cancer holistically and in understanding how cancers affect the body and understanding this interconnectedness between cancer and different organs. And then the insights you get from this can be used for theranostics, either to do things like radiotheranostics or small interfering RNA delivery or photoimmunotherapy. And just, of course, none of this work would be possible without some fantastic colleagues. So This is Jesus Pacheco Torres, who's back in Madrid, who did a lot of the PD-L1 siRNA work, the cancer fibroblast work. This is Jifu Jin, who does all our photoimmunotherapy work. Rajkumar Sharma, who does all the high-resolution extracts. Balaji Krishnamachari, our molecular biologist. And Paul Winard and Marie-France Benet, who did a lot of work with cachexia. Samda Kakad, who did a lot of work with the collagen imaging. And of course, our funding support. And I've been asked to put up this slide to please request you to provide a survey of this seminar. So thank you for your attention and happy to take questions.
Great presentation. Thank you so much for sharing your experience with us. Definitely cutting edge technology and a great vision for the future. I'm always impressed to hear your talks. The challenges that we have is in early detection, right? And we've struggled to find one technique or a combination of techniques, and we've always thought about the multi-parametric tools. It could be spectroscopy, it could be hypoxia, it could be a combination. And it's hard to put this all together without the use of AI. And we talk about the use of AI, and you alluded to it. Where do you see us heading with AI? How far away from really having useful AI tools that we can bring to clinic? And the second part of this question, where do you see the marriage between imaging and liquid biopsies? And because there is a lot of interest in, even before imaging, are there blood biomarkers that can help us, identify early cancer? I know this is a complicated question, but you're a visionary, and I wanted to pick your brain where we're heading with imaging.
So, Ihab, I can only respond with what we're doing. So, with spectroscopy, as I mentioned, we are looking at plasma spectra, and with like totally blinded samples, we could detect PDAC with 83% specificity and over 90% sensitivity. So that, I think, excited Mike Goggins very much. And then our other oncologists saw this. So for instance, Yasser Ged, we're starting to work with him with renal cell carcinoma and with detecting response to cancer immunotherapy using this approach. And again, I think their neural networks can play a role in monitoring response to therapy. And of course, with Malcolm Brock, we're looking at non-small cell lung cancer. So, so that's the spectroscopy part. And I was just telling someone about, some years back, there was this fossil test where they were saying that they could detect cancer with change in water line width. And there was this lot of excitement about using that because, plasma tests are so easy to do and everybody's looking for liquid-based biopsies, but didn't pan out. So like Peter said, don't be like fossil when I told him that we can detect PDAC with spectroscopy. But I think that is one area that can evolve, not only for cancer, but also for things like Alzheimer's, for instance, or infectious diseases, because anything that is metabolically active is pumping metabolites into the plasma and altering it. And of course, you would have to train your neural network for that specific pathway or condition and then use it. So I think that's where there is a role separate. And then, of course, for imaging, again, the same things apply. You can use it for detecting response to therapy or looking at identifying cancers. So, and for your second question was about relating biopsy and imaging. So again, as an example, when we took this NIH's NLST database and took the H&E stain slides from them and ran collagen fiber imaging on that. And we were able to say that, yes, stage 1 non-small cell lung cancers that go on to recur have high collagen fibers. Now, as you know, stage 1 non-small cell lung cancer, they cut it out and then 5 years later, that's it. And then 30% of them go on to recur. So if you can know early on that, yes, these stage 1 non-small cell lung cancers are likely to recur, you could be more aggressive with treating them. So those kinds of approaches, I think, where you can combine biopsy and imaging to closely monitor those individuals that show up at biopsy with some risk are, I think, important. So does that answer? Yes, Nat.
Thank you, Zaver. So your team has pioneered the collagen imaging with the second harmonic imaging, and it's only ex vivo, right?
Yes, the second harmonic is ex vivo.
So do you envision any advances in the in vivo technology to be able to see the collagen fibers?
So of course you know, as we were discussing, there are MRI techniques. I think Debbie Burstein had come up with collagen imaging based on saturation transfer. And then, as you were mentioning, elastography. So those are techniques where you could use MRI. And then Peter Caravan and Jenny Yang are developing probes for collagen imaging that, again, you could use for MRI. That sort of resolution where you can look at fiber orientation. You can just come up with some quantitative estimate of collagen. What we're seeing with fiber imaging is that in the aggressive breast cancers, the fibers are like long and thin. And whereas in normal tissues, they are like wavy and very different patterns.
So the structure is totally different. Okay. Yeah, I don't think we can do it in
No,
vivo, so.
no. But if you just want to look at collagen, there are probes being developed that will become available with time.
And those MRI, I think it's both MRI and PET, right? Because you can do it
MRI
maybe
and
with
PET.
gadolinium
Yes.
and also with the F18. Okay,
right.
great. Thank you.
Thank you so much for your talk. That was really just such interesting data that you showed. I had a question relating to your PD-L1 imaging. As you know, the standard is for the pathologist to come in and confirm PD-L1 staining expression for the use of those checkpoint blockade therapies. Have you compared the sensitivity of that PD-L1 imaging to current pathologic methods to detect that? And along with that, the histology to look for that PD-L1 isn't necessarily always the strongest to indicate those patients that will respond to immune checkpoint blockade. Have you considered or examined if your use of high resolution imaging with the fiber imaging or vascular imaging combined with that PD-L1 detection might provide any sort of advantage when used with AI to detect responders? Excellent. Thank
Yes,
you.
of course. So Sridhar Nigbagada, she's my colleague at Hopkins, has developed a peptide-based F18 PD-L1 imaging agent that I think was in non-human primates, and then they did a first in human, and we hope to bring that into human trials fairly soon. But what Sridhar has shown, and I think he published that in JCI, is that you can relate what you see histologically, and he shows receptor occupancy, that you can relate it to what you see with imaging. So it's a fairly good reflection of the pathology, a correlation with the pathology with the in vivo imaging. And your second point, I think you want to know if vascular imaging and other approaches, yes. So if you were to see, and I'm just guessing this based on what we have seen with hypoxia, that if you were to see regions of poor vasculature that would most likely be hypoxic, it's possible that those regions have high PD-L1. So you could possibly, but again, it has to be validated, but they could be surrogate markers.
Very nice. I have a question on the machine learning aspects. How many different types of models did you try other than the neural network? And do you have any insights into which metabolite is the most important for these models to classify? There are techniques to get at that. So,
the neural network is all done with human plasma samples. And I think we needed something like close to 80 per group for training. And then we ran about 50 blinded samples.
And do you have the idea of which in the serum?
Yeah, very good question. Yes, so I drove my poor AI person crazy saying, I want explanation of why it's picking it up. So yes, so he came up with the dominant regions in the spectra that were driving the differences. And I think there are 10 metabolic clear regions that include the usual suspects of glutamine, glucose, lactate, alanine. So all of those showed up as dominant regions driving the differences. Yeah. Right. Thank you very much for your attention.